I am going to talk about protein structure. Why?
amino acids المختلف.
Why?
خد الامينو اسيدز المختلف.
How? Why you want to study protein structure?
ممكن تبدأ function.
Because the protein structure plays a key role in its
having different functions.
jobs. وبيلعب لازم أعرف شو structure
Yes? عشان تعرف function. Is protein or our proteins have important functions?
Yes.
Like what?
Enzymes.
Enzymes. What else?
Metabolism.
Metabolism. What else?
Structure.
I will say one sentence about proteins. You are what your proteins are. You are what your proteins are. If they are good, healthy, functional, then that will reflect on your health. Everything you do is associated with a functional protein. Any motion, any action is related to proteins. So we want to know more about these functions of proteins, how they do it. In order to understand that, you have to understand the structure of proteins. You already started knowing the structure of the monomers of proteins, which are the amino acids. Understanding the structure and physical properties of some of amino acids will help you to understand the structure of proteins. And once you understand the structure of proteins, okay, you will appreciate the function of proteins and you will understand different functions of proteins. Okay. Proteins could have 4 levels of structures. The first level is the primary structure of proteins. And it is simply the amino acid sequence of in any polypeptide chain, in any oligopeptide, in any protein, the amino acid sequence is the primary structure of that protein. And what do we mean by amino acid sequence? The position, the exact position of an amino, any amino acid, where is it located? What is the order of its position? Is it the first, second, fifth, 100th, nth? These are the exact positions of amino acids. Now remember that amino acid composition is different from amino acid sequence. What is the difference? تفضل. The difference. Amino acid composition of a protein is different from amino acid sequence of that protein. What that means? How you explain it?
Because we have only 20 amino acids, the way we change between any proteins is not like glycine is مثلا موجود في كل البروتينات, but its place in each protein is different. Its placement changes the protein. هاي هي sequence. أما composition هو number or types of amino acids that goes into protein.
Yes, you are right. Amino acid composition. How many amino acids of each amino acid in that protein? What is the percentage of each amino acid composing that protein? But the amino acid sequence, what is the location of a specific amino acid in that protein? Okay? Location is very important. Amino acid sequence or primary structure is very important because it will determine the function of the protein. And I will give you some examples in which a change of one amino acid in 500 amino acids protein could lead to non-functional protein, and that could lead to death. Okay? And we will discuss that. A second important level of protein structure is the secondary structure. Secondary structure describes the peptide backbone conformation. Yani how it looks in a three-dimensional structure. The peptide bond backbone, the peptide backbone, how it looks in a three-dimensional structure. It looks in different ways. The three-dimensional structure of the backbone of the polypeptide chain looks in different ways. It could look in a helical structure, it could look in a sheet structure, it could be a tear, it could be bent, it could be unordered structure. So different types of shapes, okay, the backbone of the polypeptide could appear in the three-dimensional form. And we will talk about these, how they are formed, and why they are formed. The third level of protein structure is the tertiary structure. Tertiary structure is also the three-dimensional structure of every atom in that protein. And that happens by when the secondary structural elements fold on each other, okay, they will form a spherical, compacted, global structure of that protein, and that protein is called the folded protein. And remember that each protein must be folded correctly. There are millions of probabilities that a protein will be folded, but only one type of folded is the stable one, and that type of the folded protein or that type of tertiary structure is the functional form of that protein. If there is any mistake in the folding of that protein, the protein will lose its function. So folded proteins are very important to be folded correctly. And what determines the correct folding of a protein is the primary structure and secondary structure of those proteins. A fourth type of, or fourth level of a protein structure, which is not necessarily for some proteins. Some proteins require quaternary structure to be functional, and some other proteins do not require any quaternary structure for function. What we mean by quaternary structure is simply an aggregation of more than one subunit or more than one polypeptide chain in order for that protein to be functional. Yani, for example, hemoglobin is composed of 4 polypeptide chains. One polypeptide chain will not make hemoglobin functional. 2 polypeptide chains, no, not functional hemoglobin. Hemoglobin requires to be in a tetramer or in a aggregation of 4 subunits. Other proteins require to be aggregated into 2 subunits as dimers. Some other proteins do not require to have a quaternary structure. Just one subunit or one polypeptide chain of that protein will do the function if it is properly folded. Okay? So what is a quaternary structure? Well, this is an example. What is a quaternary structure? Aggregation of more than one subunit or polypeptide chain. An aggregation of more than one subunit. Tertiary structure is the folded protein, the correct fold of a protein, how it looks in 3 dimensions. Every bond, every atom, how it looks in 3 dimensions. This is the primary structure. Very simple. What determines a protein, X protein, to have this primary structure, while Y protein to have this different primary structure? Why primary structures of different proteins are different? They are not the same. Why?
Function.
Because they have different sequence of amino acids.
Why they have different sequences? Although we have 20 amino acids that make proteins, but what makes primary structure of different proteins? What makes them different?
Because of the function of the protein. Function of the protein.
Function of the protein. Excuse me? وما الذي يعمل الاختلاف؟ Genes. Genes. Okay? The gene, every polypeptide chain has a gene responsible for its synthesis. When it is transcribed, messenger RNA will be produced, okay, according to the coding sequence in the gene. And that will be translated into amino acids and proteins. So primary structure of different proteins is different because the differences in the genes. What is a gene?
It's a part of the DNA.
Part of the
that
DNA
codes for some-
that have specific sequence of nucleotides. Okay? Specific sequence of nucleotides. Piece of DNA with a specific sequence. This is the secondary structure. This is an example of secondary structure. It is a helix, helical-like structure. We will talk about it. So the amino acids in the secondary structure do not look like a straight chain or a zigzag form of chain, but it looks like a coil. Okay? Like a coil. And we will talk about these types of coils. From the correct amino acid sequence, a correct secondary structure will be produced, and according to these secondary structures that comes from primary, a tertiary structure will be formed. This is the folded protein. The spherical packed protein is the tertiary structure of a protein. And this is the quaternary structure. Aggregation of more than one polypeptide chain like this will produce a quaternary structure like this, 1, 2, 3, 4, okay, in order to be functional. Some proteins do not require an aggregation of more than one subunit. One subunit will be sufficient and it will be functional. Okay? This is how a peptide bond is formed. And it is formed, as you see, this is the alpha carbon, this is the alpha amino, and this is the alpha carboxylic group. Okay? This is alpha carboxylic because it is attached to the alpha carbon atom. This is alpha amino because it is attached to alpha carbon atom. Okay? Every amino acid must have alpha amino and alpha carboxylic and hydrogen and an R group. Now, this is one amino acid. In order to make a peptide bond, there will be a condensation between two amino acids. This is the alpha amino of the second amino acid, and the alpha carboxylic of the first will be condensed with the alpha amino of the second amino acid. Water will be produced. A bond will be formed between this carbon and this nitrogen, forming a peptide bond like this. Okay? There are many peptide bonds in any polypeptide chain. Okay? What is the number of peptide bonds? أقل من الأمينو أسيدز واحد. فلو كان عندنا 100 أمينو أسيد يكون عندنا 99 peptide bonds. Okay? لو كان عندنا 2 أمينو أسيدز شو بنسمي الستركتشر اللي طلع لنا؟ داي بيبتايد. 3 أمينو أسيدز 4 10 أوليغو مئات. خلاص انتهينا. شو معنات ريسيديو؟ amino acid within a protein is called amino acid residue. طيب. This is a presentation of a polypeptide chain. For any polypeptide chain, there is a beginning and there is an end to it. Like a gene, it has an beginning. What is the beginning of any gene? What is the end? And what is the beginning of a protein? N-terminal. And what is the end of any protein? C-terminal. ليش سميناها N-terminal؟ من وين أجت الأمينو جروب؟ من الفيرست أمينو أسيد. So always the first amino acid has a free alpha amino group, and that end is called the N-terminal. And the last amino acid has a free alpha carboxylic group, and that is called the C-terminal. Reading of any amino acid sequence for a protein starting from the C-terminal to N-terminal. Okay? N-terminal سيدا؟ Yes. أنا حكيت خطأ، خصوصي حكيت خطأ عشان أشوفكم معي ولا لا. فالدغري انتبهتوا، برافو عليك. So reading start from N-terminal to C-terminal. This is amino acid number 1, 2, 3, etc., etc. Okay? Understood?
Yes.
Very good. Very good. Very good. الآن بدنا نتكلم عن characteristics of peptide bonds. Peptide bonds like double bonds. They are rigid. No rotation of these peptide bonds like double bonds. They are formed in planes. And each one of these represent a plane of a peptide bond that is composed of 6 atoms. This is 1 atom, 2, 3, 4, 5, 6. These are the 6 atoms that compose the plane of a peptide bond. The plane of the peptide bond is rotating, while the peptide bond itself is rigid, not rotating. Our groups are rotating around the alpha carbons. Planes are rotated around the alpha carbons. But the rotation of these planes is limited. Okay, there are specific angles in which this plane rotates around the alpha and this plane around the alpha carbon through the C alpha and NC alpha. These limitations will produce, okay, different types of secondary structures, as you will see. Remember that the peptide bonds are in a trans, and it is important to be in a trans because when it rotates, the planes, when it rotates, to avoid the hindrance, okay, between the R groups and part of the peptide bonds. What is the importance of primary structure? This is the answer. Did you understand the answer? What is the answer?
Function.
What is the example here? Hemoglobin. This is hemoglobin. It is composed of, it has quaternary structure. It has 2 alpha chains and 2 beta chains. Later on, we will not name them as chains, subunits, and I will tell you why at that time. Okay? The 2 alpha chains and the 2 beta chains have a collection of 574 amino acids. 141 for alphas multiplied by 2, and 146 amino acids for each beta, totally 574. It was found in a disease that in beta chain, at position 6, valine replacing glutamic acid. If the 2 beta chains have the same problem, then we have 2 amino acids, and these 574 amino acids have been changed from valine, from glutamic acid to valine. Is there any difference between glutamic acid and valine? Huge difference. Glue is charged polar, okay, while valine is hydrophobic, non-polar. وهذا الديفرنس عمل changes في البي اي. البي اي originally 6.87 عشان هاي التو changes تحولت ل 7.09. لأنه هذا hydrophobic وهذا charged to polar, okay, that caused an aggregation of these hemoglobin molecules into this fiber-like structure. Now remember, hemoglobin is found soluble in red blood cells, carrying oxygen from lung to tissues and CO2 from tissues to lung. Now, and it is soluble, while in this case, when it is aggregated, it becomes a fiber-like structure, and it is insoluble. And if it is insoluble, it will affect the membrane of the red blood cells. And instead of the red blood cells to be in a concave structure, okay, it will be in a sickled type structure. So these cells become very fragile, they will break, and as a result, anemia will be produced. In addition to that, these sickled cells will block the capillaries and prevent blood carrying oxygen to reach the tissues. What will happen to the tissues? The tissues will, of course, ischemia, no oxygen, nothing, and oxygen is highly required, okay, for energy production, as you will see, okay? So if there is no oxygen, then the tissue will die. And this is a result. Death of tissues, brain, kidney, anything, is a result of a change of one amino acid in the primary structure of one of the components of hemoglobin, in beta globin chain. So primary structure, as I mentioned earlier, it determines the function of the protein. If it is wrong, protein will not fold it correctly and it will be non-functional. An example of this is the sickle cell disease. Sickle cell disease, okay, resulted from changes in one amino acid that instead of a glue in the beta chain. If one beta chain has this problem, there will be no problem in this disease. Sickle cell trait. It is a heterozygous. But if the two beta chains are defected, then the person have a serious issue. Okay? Sickle cell disease. And that is very serious. This is the importance of primary structure. How it determines the function of the protein. There are a lot of genetic diseases in which one point in mutation in their genes resulted in changes in the primary structure. And changes in the primary structure will produce non-functional proteins. Questions? The second level of a protein structure is the secondary structure that results from the primary structure. There are specific groups of amino acids that will produce a specific type of secondary structure. Supposedly we have a polypeptide chain extended from this region to that region. Okay? In this region, we will see some amino acids will produce, they will be oriented in 3 dimensions to produce a specific regular structure like a helix. And that specific regular structure coming from this type of these types of amino acids is called helix. While in this region of a polypeptide chain, there are other types of amino acids that will produce other type of secondary structure. In this part of the polypeptide chain, there are other types of amino acids that will produce no regular structures, unordered structure. And there, between this and this, there is also some other types of amino acids that will produce a fourth type of secondary structure called tails, for example. So we have different forms of regular secondary structures produced, okay, because of the types of amino acids and how those amino acids are oriented in 3 dimensions. What are these types of secondary structural elements, regular secondary structural elements? Alpha helix, beta pleated sheet, tails, and loops. Questions? So, this is the alpha helix, one of the regular secondary structures. It could be, as I mentioned, right-handed or left-handed. If it is the coiling or the twisting clockwise, then it is right-handed. Counterclockwise, left-handed. Okay? L-amino acids are doing the right-handed alpha helix. D-amino acids do not make any helices. So we don't have D amines. If you mix L and D together, you will get no secondary structure elements. Now the question, 2 questions. What stabilizes this regular structure to be there always in the protein to make the tertiary structure? And the other question, what type of amino acids that make this regular structure? تفضل.
المولكيولز اللي are stabilizing in hydrogen bond.
هيدروجين بونز بين مين وبين مين؟
In between the R groups, are common acids.
Are groups
Common acids.
of amino acids. Good. Very good. Yes, half of the answer is correct and the other half is not.
hydrogen bonds not
Hydrogen bonds, yes, you are right.
The hydrogen.
But our groups have nothing to do with hydrogen bonds in secondary structures.
Between the oxygen and hydrogen.
الكربوكسيل والامين الاو على الكربوكسيل والاتباع.
Yes. The hydrogen bonds will be formed between the nitrogen of a peptide bond with the carbonyl oxygen of another peptide bond. Not the next peptide bond. Not the next to the next. Not next to the next to the next. But 4 amino acids far. So there will be a 4 amino acids gap between that hydrogen bond, between that amino acids and 4 amino acids in front. With the 5th one, it will do the hydrogen bonding between the nitrogen of the peptide bond and the carbonyl oxygen of the peptide bond with amino acid number 5. 2 with 6, 3 with 7, 4 with 8, with 9, etc. This is how these hydrogen bondings are formed. Okay? 4 amino acids away. That will stabilize this type of structure. Our groups, as you see, they are extending away. This type of interaction is called short-range interactions. Short-range interactions. لأنه في interactions between neighboring amino acids. Number 1 with 5, number 2 with 6, etc. While in tertiary structure, as you will see, there is a long-range interaction. There will be interaction between amino acid number 5 with amino acid number 600. these are long range. شو اللي جاب نمبر 5 بجنب ال 600 امينو اسيد؟ فولدنج، لانه صار correct folding فصار long، صارت هاي الانتراكشن that will stabilize the structure. These are numbers not necessarily to memorize them. Okay? This is how it looks when you look from this direction, from up to down. This is the alpha helix. And these are the amino acids that do not favor the formation of alpha helix. دكتور حكيت إنه there are specific types of amino acids that like to make alpha helix or beta sheet or beta tail. Okay? By statistical analysis, it was found that glycine is a strong broker of alpha helix. Why? لأنه ال R group is very small, okay, were unable to make these coiling. So it is a broker for alpha helix. Proline is a broker also because of this secondary nitrogen that cannot make hydrogen bonding with any carbonyl oxygen, and also cannot rotate the NC alpha rotation. Okay? Close proximity of amino acids that have the same charge. Give me an example of amino acids having the same charge. So well? Yes. You will never see, for example, histidine and lysine together. You will never see lysine and arginine. You will never see asp and glue. Okay? Cannot make this coil. So they are brokers. Also amino acids with a big size of side chains, okay, also do not favor making the alpha helix. So, what are amino acids that favors making the alpha helix? Hydrophobic amino acids. Okay, the hydrophobic amino acids. Usually, in a stretch of amino acids, if you want to tell if this stretch, another stretch is going to make alpha helix or not, you must look an array or series of amino acids that are hydrophobic. Usually, the first amino acid is positively charged, and the last amino acid in that stretch is negatively charged, and between all hydrophobic. This you could tell that here is an alpha helix in this protein. Okay? بدكم نوقف هون ولا نستمر؟ بدكم نوقف؟ ليش؟ كل الشعب وصلنا للبيتا شيت. احكي كلمتين عن البيتا شيت. البيتا شيت عبارة عن strand like this. It is not coiled ولا helix. Okay? It is very, very small coil. and it is in a zigzag form, trans for the R groups. Usually, many strands of that protein, of any protein, will make structures like this. And a hydrogen bonding will be formed between amino peptide bonds in this strand with another peptide bonds, comparable peptide bonds in the second strand. And it could be many strands, not only two strands, okay? And those strands could be parallel or could be anti-parallel. معنات parallel يعني N-terminal مقابل N-terminal، C-terminal تبع هذا الستريتش مقابل C-terminal، ومعمول بينهم هيدروجين بونز. الانتي-parallel بتكون N-terminal مقابل C-terminal مقابل N-terminal C-terminal، اوكي؟ وعاملين هيدروجين بونز وبيعملوا شيت لايك ستراكشر، عشان هيك بيسموهم بيتا بليتد شيت. بيتا بليتد شيت. أقف لهذا الحد ونكمل المرة القادمة.